Welcome, everyone, to the Spotted Lanternfly in Massachusetts quarterly update, February 2024. Thank you for spending your Valentine's Day morning with us. I am Jennifer Foreman-Orth. I work at the Mass Department of Agricultural Resources. I am here with Astra Perkins and Elizabeth Barnes, and we're very pleased today to welcome Melody Kenna, who is a research entomologist at the U.S. Forest Service. We're covering a few different topics today. First, Melody will be speaking to you about temperature tolerance in spotted lanternfly. Following that, Astra Perkins will give an update on what's going on in Massachusetts with spotted lanternfly survey and treatment. And then Elizabeth will have a brief presentation about what to look for right now in terms of detecting spotted lanternfly, followed by as much time as we have for questions and discussion. And that's pretty much it for me. I'm going to turn it over to Melody. Are you ready, Melody?
I am ready.
Excellent. You should just be able to go ahead and share your screen.
Okay, and go ahead and start.
Yeah, we should be all set. Looks good. Thank you.
All right. So today I'm going to share with you a compilation of published information, unpublished data, and some papers that others have published. I will tag those as we go through. I'll show you how to watch for that. So first, I'm going to give you a real brief overview of this insect. Spotted lanternfly came from Asia. It's found in China, Bangladesh, and Vietnam. And if you look on the right side of the screen here, you'll see the areas in Asia where it's found. And then I have red lines that come across to North America, and you can see what part of North America that equates to in terms of latitude. It's also invasive in South Korea. In the US, this is the most recent map that's been published of where it's found. 17 states with infestation, 7 with interior quarantines. There's no federal quarantine for this insect, and it spreads both naturally and by human-aided transport. So meet the insect. The adult is here on the left of the screen. The egg masses that they lay are in the middle. And on the right, you'll see a newly molted 4th instar nymph. This insect has a 1-year life cycle. This life cycle that I'm showing on the screen is based on the core area where it was first discovered in Pennsylvania. And so those timings are appropriate for that area. But if you go to cooler or warmer areas, those timings can shift. For example, the hatch can shift as early as March or later in June and can continue into July in some places, just as an example. As I said, this insect does hitchhike quite readily, all stages, adults, nymphs, and egg masses. They can be on something that we move as humans from place to place, or they can be on the vehicles that we either move in ourselves or move things around in. So because it can hitchhike, one of the things that we wanted to know with reference to temperature was how long is the mobile stages of this insect going to be able to survive while in transport. And as you can see from this graph, survival time is on the y-axis and temperature is on the x-axis. Later in this presentation, I'll give you some walks between Celsius and Fahrenheit. But basically at summer temperatures, they don't live very long without food. But as the temperatures are cooler, which is more during the time frame when the nymphs might be present, they can live quite a while, especially the first instars. If they end up on something shortly after hatch, they can live a longer period of time at the cool temperatures in the spring and have probably long enough to find food once they get to a location. Adults, however, survive less than a week without food across a broad range of temperatures. We're also looking at what kind of high-end temperatures might be used to kill the egg masses that appear on either cargo or vehicles. as a possible way to handle them for things that are moving across straight lines. This is the preliminary data on it. We're finalizing this more here in this next year or two. But preliminarily, it looks like that if you give them 50 degrees C, which is 131 degrees Fahrenheit, for more than 30 minutes, you're going to kill almost every single egg in the mass. This is actually less than the temperatures that are required to heat treat solid wood packing material to kill things like Asian longhorn beetle. But we'll finalize this more. We're also looking at it for spongy moth, which it would be nice if you could kill the egg masses of both with the same treatment, and it appears that that might be possible. So this insect, there's been some modeling done, and if you watch for these purple little messages somewhere on the slide, that'll tell you where this data is from. This happens to be from somebody else's published article. Just watch for the purple, you'll see where the data is from. But in this model, it shows that the lanternfly can potentially establish and develop across quite a range here in the eastern half of the US, as well as parts of the Pacific coast. And there are several factors involved in that, but just look at where it might be able to establish. If we start with eggs, if you're not familiar with this insect, it will lay its eggs on almost anything. I haven't shown you the rusted tin cans that we often find them on in the forest, but they'll lay on almost anything. I'm showing here some birch bark in the lower right corner, a rock in the upper right corner, and the left side is a tree, and I've shown you some arrows to the undersides of these branches where they're just loaded with egg masses. This is typical where they will lay. So to kill egg masses in nature, what are the winter conditions that are likely to kill them? There's still some work going on to determine what the supercooling point is and the temperature at which, if they're exposed to that temperature, everything's going to die, and it doesn't have to be exposed for long periods of time. That work is still going on. But this graph shows you that if you get mean daily temperatures in January that average somewhere around -4 Fahrenheit, which is -20 Celsius, then pretty much all the eggs are going to be dead. And it's a more or less linear relationship. So the colder the January, the more likely you're going to have the eggs not survive. And then based on that graph, this is projecting it across the landscape. This is some work I did with somebody in Canada that was interested in what it might do in Canada. You can see that here in New England, for the most part, we're not going to get huge amount of winter kill until you get to the northern parts of New England. Talking about killing egg masses with high heat or low temperatures, that's survival thresholds. So I'm going to just kind of give you a little hint that we're going to talk about two types of thresholds in the talk. Survival thresholds are temperatures at which mortality approaches 100%. And they can be either freezing lower temperatures or higher temperatures that disrupt the metabolic processes and exhaust the insect's resources. Developmental thresholds are temperatures at which immatures can molt or eggs can hatch. So this is their normal temperature range over which they can develop or grow. And these are normally estimated at constant temperatures, but that can be problematic, as I'll show you later. So developmental threshold for eggs, the lower threshold is 7.39 Celsius, which is 45 degrees Fahrenheit. That's the lowest temperature at which the eggs will develop. But they can hatch between 10 degrees and 25 degrees C, which is 50 to 77 Fahrenheit. And the upper threshold is most likely about 25 degrees C. This is newly laid eggs. We don't know yet exactly how older eggs will respond to some of the higher temperatures. And the eggs go through what is called a diapause. And so there's 3 phases to diapause: pre-diapause, diapause, and post-diapause. And each of those, we believe, have different thermal responses. We're still trying to understand those. But the picture here on the right is actual nymphs hatching. They first hatch. They're quite white, but they can color up quickly to their black and white spots. And the data on eggs have been used as well as nymphs and adults. So there are websites that you can access online. And you can see the addresses here on this slide that will let you predict for whatever you want to look at, what stage they're going to be at a particular time. And this was April 25th. You can see there were parts of the range where hatch hadn't occurred yet, but the warmer parts where they should be already hatched and growing. And even in this particular one, although they're not in Florida, I was predicting there might be some adults already. You can also predict hatch time. This is another website that you can go and look at. So one thing that we did, though, is we wanted to see if there was variation in their responses to temperature across their range that they currently are in. So we collected from 20 different sites that were in different plant cold hardiness zones across the range and did some evaluations of them. We held the eggs at a constant 15 degrees C, which is a temperature at which they will hatch. And we found that the variation had an interesting pattern, that the cooler sites and the warmer sites both took longer to hatch than the sites that were what we would consider equivalent to the core area of where they were first introduced, which is the 6B cold hardiness zone. That's the green slice here. And what we think is going on is that obviously in a cold site, if they hatch too early, they risk being frozen. And if they're in a warmer site, if they hatch too early, they risk running out of resources before the adults can lay. And the reason that is because the laying of eggs is timed not by temperature, but by some light cycle parameter that we don't know exactly yet. There's variation across the range, and here's the actual first reports of hatch observations. You can see that it isn't following precisely a north to south gradient. So all the south ones don't hatch first and then the northern ones. So there's something besides temperature going on that is affecting this. And since we know that there is variation, there could be adaptation to particular locations happening already. We did in Fairfield, Connecticut, last year watched the egg hatch over time. We're going to repeat that this spring as well. Here in Connecticut, they started hatching about the beginning of May, but there were individuals still hatching the first part of July. So this is a very spread out hatch for this insect. And the early hatchers, a lot of them were on rocks that were in sunny locations. So they were hatching on these rocks that got extra heat long before they were hatching on the tree boles. And at this point, there's going to be a poll question.
All right, folks, here's the question. What are the upper and lower temperatures at which most spotted lanternfly eggs will die? Looks like most people answered B. Melody, do you want to weigh in or add any additional discussion here?
I will just roll forward and show them that that is the correct answer. B is the correct answer. There's the full answer. Minimum January temperatures that are less than or equal to -20 C, which is -4 Fahrenheit, and with exposure to 50 degrees C, which is 131 degrees Fahrenheit for more than 30 minutes. That is the correct answer.
So we had 70% of the folks get it correct. All right, keep going.
All right, so we've talked about eggs a bit. Now we're going to talk about the mobile stages. If you're not familiar with this insect, the picture on the left is some first instars all lined up along the stem of a tree of heaven. The next two pictures is a fourth instar on the bottom and a fourth instar that is in the process of molting on the top. And then you have a second or third instar on the top in the next panel, a newly molted adult and its skin, and then a couple of nymphs. So these are the mobile stages. We're going to talk about temperature in the mobile stages now. So as I told you, the developmental thresholds, we usually estimate them using constant temperatures. I've given you the Fahrenheit equivalents down in the lower right corner. So for all of the mobile instars, first and second instars have minimums around 12 to 13 degrees C, and thirds and fourths 8 to 6 degrees C. The upper thresholds we've estimated for first to be out around 40 plus Celsius. But for the other instars, 35 degrees based on these data. And you'll see later that that might not be completely accurate for the upper thresholds. Here's just an example of survival at the different temperatures. If you look at the brown, that's 40 degrees, less than a week survival for first instars at that temperature. And this is with food. 5 degrees, some of them survive out to about 16 days or so. And you can see that they do the best at 30 degrees, 25 degrees, 20 degrees. That's their preferred temperatures, but they will survive quite well at 15. They survive so-so at 35. So I've got the information that I already showed you on the far right, that the lower thresholds are, in those red circles that I'm showing for first and second instar, and upper thresholds for thirds and fourths at the bottom. So we did some checking to see if this was accurate or not. We exposed the insects to temperatures below the threshold in combination with the temperature in the preferred range. So, for example, first instars, we gave them 5 degrees C and 20 degrees C, and seconds the same. And then for the higher instars, we gave them 40 degrees with 20 or 35 degrees with 20. And what we found is that when these temperatures below or above the predicted thresholds were included with ones that were permissive, they were able to survive and molt. So even though the upper thresholds might be 35 degrees C, if they've got an alternating temperature regime where there's permissive temperatures involved, they are still able to molt. So that means that they could potentially go further north and south, where temperatures are in those what we consider not preferred zones, and do be able to develop. And this was an additional study that we're just finishing now. We provided these insects with a chamber in which we had mimicked the Stockton, California temperatures from 2022. So we took the average high and low temperatures for that year, and we held the insects for a week at whatever that average was. The bottom shows you the temperatures. And we introduced them into this regime in March because that's when we predict that the hatch will occur. And they developed quickly through the instars, ending up having adults as early as the beginning of June, which is comparable to what we're seeing in North Carolina. And then the adults survived a really long time, and they laid a lot of egg masses. Females laid multiple egg masses, and we even had some precocious hatch, some hatch that occurred before winter actually had come into play. So we don't know exactly what that is going to mean for this temperature zone, but interesting finding nonetheless. Also, I've circled in red, there was a week that we set the chamber temperature at 40 degrees C for the high temperature, and it was about 20 degrees C for the low temperature. And you can see above that it really didn't have that big of an impact on survival of adults, and they were actually laying eggs during that period of time. So they can survive these summer heat waves and do just fine. We also looked at the responses of those eggs that we had hatched from those 20 different populations at 2 temperatures, at 15 degrees C for the first instar, and the next slide will be 25. You can see that there's substantial variation across the invaded range, and even within populations, in how long it takes a first instar to develop to molt to the second. So there's a lot of variability that may be able to be acted upon, and adaptation to particular climates may be occurring. And I'll show you some evidence that it already is occurring here in a second. At the higher temperatures, there's a lot less variation. And none of this is tied directly to cold hardiness zone. So hot locations didn't necessarily grow faster than cooler ones in this particular data set. But in the previous data set that we did, where we compared some New Jersey, Pennsylvania, and Virginia egg masses, where the Pennsylvania was the warmest site, you can see there's quite a bit of difference. And what this means, all these differences mean, is that the lower thresholds is going to vary between populations for different instars, as much as up to 4 degrees C, which can have a substantial impact on the development speed of the insect. The other thing that happens is you can see the black lines for Pennsylvania cross the other lines. This is evidence that there is genetic basis for this difference that we're seeing, and that adaptation may have already indeed occurred. So let's move on then to the adults. And I'm going to give you a fun little video here. This is the adults with their projectile honeydew. You can see going on there in that photo. And the other pictures are trees that are heavily infested, and the black sooty mold and the slime fluxes that are associated with them. Here's some pictures of other pictures of adults. You can see these females with the bright yellow band along their abdomen. Those are the females that are mature and ready for egg laying. With the adults, Dennis Calvin at Penn State has predicted that it takes about 1,500 to higher levels of degree days at base 10 for them to reach the point of egg laying in the fall. So he projected then across the landscape where these would occur. So any place on the left-hand map that goes to the light yellow phase, that's where adults are going to have plenty of time to reach the egg laying stage. But you can see at the bottom, North Carolina, for example, they've got more than enough. And that's where I was telling you that they're going to live longer, they're going to lay more eggs, and they're going to die of old age. Whereas in the north, you're going to have temperatures that intervene and will kill them. And we're going to talk about that in a second. The picture on the right is the actual first sightings of adults. You can see North Carolina, June 30th, but I believe that in a study that was going on down there, it was closer to the beginning of June. And then Connecticut in July for first sightings of adults for 2023. First sightings for egg masses, however, is a different story. Across the entire range, it's only a little over a week difference in when first eggs were observed. You can see on the left-hand side, and first adult sightings I've left up on the right-hand side. So the adults in the south are there a lot longer before they start laying. And so they can get much larger and potentially lay a lot more. So we think that there's a light cycle trigger that is triggering laying, because it happens at about the same time across the range. If it was temperature alone or maturity of the insects, it would be varied across the range. In Connecticut, we looked at oviposition, and this was the lower 5.6 meters of the tree. So we weren't actually assessing what was being laid up higher. We know that their egg masses laid up higher. But the first egg masses were laid September 21st or 26th, and all the adults were found dead November 23rd or 21st. We were just checking once a week. And I'll show you what the killing temperatures were in the next slide. The peak time of egg mass laying in the 2 years was slightly different, October 24th versus November 8th. But what happens with this insect is when it gets too hot, the adults go down where it's cooler in the shade to feed, or when it's too cold, they move down lower again to try to stay warm. They will actually get down in the duff around the base of the tree when it gets particularly cold. but the killing temperatures in both years, you can see the orange arrows point to temperatures that drop below freezing. So there were 3 occurrences of freezing temperatures that occurred in both years before we actually had the hard freeze, which is the red arrow, that actually killed the adults. And it's interesting, the hard freeze actually occurred on November 21st, both years. This is when the temperature dropped to 28 degrees Fahrenheit or lower for an hour or more. That's when they actually froze. And so if the adults don't appear in the landscape until later, and then they get frozen because they're in a northern climate, a large portion of the population gets killed off and has no opportunity to lay. Whereas in the south, you don't have this occurring. And so that's what feeds into this estimate. This is also on the web. The parts of the US where there's going to be at least one individual replace the individual that was there the last year, or more than one replace. And you can see that large portions of the red, there's more than 3 replacements for every individual in the previous year. And that when you get into the purple, that's where there's less than 1. So there's definitely some mortality going on. And I want to acknowledge, if you've been watching, there are a lot of collaborators that were involved in these projects. There's a lot of technical support, as well as people in the field that have helped find sites for egg mass collection or helped us collect egg masses. There is funding from APHIS through interagency agreements, and that's my email address there. And we have our second poll question.
Okay, it's launched and we've got 5 choices here for quiz question number 2. What temperatures will kill adult spotted lanternflies? Your choices are A, temperatures less than 28 degrees Fahrenheit for 2 hours. B, temperatures for more than 95 degrees Fahrenheit for 1 week. C, 2 days at 86 degrees Fahrenheit with no food. D, both A and C, or E, both A and B. All right, Melody, 41% said E, both A and B. How did they do?
No, it looks like D had more.
All right,
Is that right? Or no, okay.
No.
41%.
Unfortunately,
Okay.
no.
All right. Let me show you. The correct answer is D. So exposure to fall temperatures of 28 degrees Fahrenheit for a couple hours, or C, a couple days at 30 degrees C, which is 86 degrees Fahrenheit without food. That's what will kill adults. And that's the end of my presentation, and I know questions will be at the end. Hopefully I've left plenty of time for that.
So right now, I think I'm gonna turn it over to Astra. Elizabeth, is that okay? Or do you have a slide or?
No, it's going to go over to Astra next.
Okay. Right. So I'd like to welcome our state survey coordinator, Astra Perkins, who is also our spotted lanternfly survey team lead, to give a presentation about current survey and treatment work with spotted lanternfly.
All right. So I'm just going to give a quick update about what's going on this winter with our survey. So if you've been to these before, you know, I kind of just do a little recap. This one's pretty quick. So as of November, during our last webinar, we had found 12 populations within the state. So those are shown in orange on this map. And prior to the end of August 2023, we only had 4. So we have found 8 in a couple months over the fall here. So, and these are the list of them, starting from the first ones that we found to the newest. So Fitchburg, Shrewsbury, Springfield, and Worcester. We knew about those in 2022, 2023. And then Holyoke, Agawam, West Springfield, Weston, Wellesley, Southborough, Chelmsford, and Ashland were all found in the autumn of this year. And I will say that a lot of those are pretty small so far, compared to the first 4. So this is our winter survey. As Melody was saying, most of the adults died off in late November. This is what they look like when they're dead and stuck to the tree. I call these guys little deadites if you're an Evil Dead fan. Their eyes kind of get this gray hue and they no longer have like an orangey antenna. Actually, I think these are their antenna, but they're usually orangey and they have like, they look dead. They look like little zombies. And we do find them attached to the trees every so often. So they can hang on real good with those legs, and sometimes their suckers are still in the trees. So right now we're just looking for egg masses. We've returned to the parcels that we've previously found adults on during the live season, and we search pretty much all materials in the area for those egg masses. So you'll see here, this is on like kind of a dead log where we have all of these. And then this is, I think this is like a bulldozer or some rusty equipment lying around in a yard. So they'll lay on pretty much anything. And you can also see in this picture that we have some hatched egg masses. So those are old and from the previous season. And then there's new ones laid on top of them. So we are out there scraping right now. You pop the eggs first and then scrape away the leftover mass. We'll go over that more later. And we use the extender pole with a paint scraper at the end to get higher egg masses. And we have poles that extend to 24 feet. So far this winter, we've scraped about 6,000 egg masses. About 3,700 of them are on 17 parcels. The USDA and MDAR are splitting up the areas to try to cover more ground. And we are seeing that the sites that we treated this year seem to have fewer than they had last year. We were able to get to those sites a little bit earlier this year. So before we kind of hit that peak of egg laying, we hit them with some bifenthrin spray, which is a contact kill, and kind of knocked down those populations before they could really go wild with the egg masses. So our next steps right now are just looking into treating again for this year. We're looking at each infestation to decide where to target. Usually that equates to where the highest density areas are of adults. And then we'll do a continuation of our delimitation surveys. And if you ever think you see one, take a picture and send it to this website. And that is all I have for right now.
Thank you, Astra. I think I'm going to hand it over to Elizabeth.
So can you see my video now?
Yes.
Okay, all right. I'm gonna hit play and then ask her if you want to talk through the egg mass scraping. That would be awesome.
Sure. Yeah, so Elizabeth came out with us and took some of these videos of us actually doing egg mass scraping. I think most of it's actually her. So she's just showing you right now the cards that we have, and then kind of how you pop all the all the eggs first to kill them, make sure they're dead, and then scrape them away. And this looks like it was just a dead log found on the ground. And then here we have a birch. This is one of our other tools that we use, are these just really cheap windshield ice scrapers. They're really good for doing this kind of egg mass scraping. And so we've seen, you know, dead log. This is maybe also a dead birch, but we'll see here that they're kind of on everything. So here's a dead cherry that they really loved. I'm sure there were also some on the live part of the tree to the right side of the screen there. But they really went ham on this kind of sheltered overhang of a dead log. Probably had the right texture and felt like protected for them so a bunch of females decided to lay there. And this is Elizabeth doing a great job getting rid of all of those. So we go out here as often as we possibly can. Really the only time we're not out is when there's like active precipitation happening. Here's another, looks like it's on a maple in a little groove. So you can see they really hide, and you can barely tell that that's there in this video. They really blend in on a lot of these trees. It's kind of crazy how much they can blend in on different types of bark. So here we have another. Just gonna pop those and scrape them. So this is just demonstrating to you guys some like good technique for how to actually destroy these egg masses. We also ask other people to, you know, if they can, scrape into some alcohol, but you know, a lot of people, you're not carrying around hand sanitizer so much anymore. So just kind of pop them. That's a great example of one. That looked like it hadn't been affected by the elements at all. Sometimes over the course of the season, the clay will get a little worn away. Just seems thinner. So that looked like it hadn't really been affected at all. And then here we have some bittersweet wrapped around a tree. And they're kind of, you can't totally see it, but they're under the bittersweet that's like wrapped around the tree. So they just really get into anywhere and try to hide these egg masses. And then we have our scraper pole. So unfortunately this day was really overcast, so it was really hard to see. You probably can't see. This is in Massachusetts, yeah. And then these trees, there's Alanthus, and this tree had over 100 egg masses on it, just on the one tree. That's a lot for what we find in most places. And again, really backlit, unfortunately, but this is kind of how it looks to us, unless we're looking through binoculars. You can see we have the pole kind of fully extended there. And yeah, these trees were just sort of like completely covered in egg masses. I'm sure if we went out there again with some brighter sunlight, we'd find a few more, but we scraped, I think, like 170 off of just these two Alanthus trees. And this is in Springfield. And you can see it's kind of hard when you get that pole fully extended to control everything. So we have to kind of go at it for a few minutes to try to get that off.
Great, thank you, Astra.
Okay, quiz question. Our final poll question is about to be deployed. So the question is, how do spotted lanternfly spend the winter? And you have 4 choices. The first would be as eggs. The second would be as nymphs. The third would be as adults. And the fourth is just in nice warm sweaters. All right, we're going to give people about 10 seconds to submit their responses to this one. I see some cheeky folks choosing D, but that's okay. I appreciate that. All right, we're at 90%. I'm going to let it go for another 5 seconds or so. Okay, I'm going to end the poll. We had 88% of folks say as eggs, which is the correct answer, which is great. 5% said nymphs, 5% said adults, and 2% cheekily answered in nice warm sweaters. So thank you. All right. I will hand it back to you, Elizabeth.
Great. So to wrap things up, I'm just going to go over some quick information about identification and also what you should do if you see a lanternfly. So this is starting out with the life stages you're more likely to see in the summer, but I wanted to share these just because, you know, in the next few months, you might start to see them. So when they hatch out, they are black with white spots, and you can see here just how tiny they are. So that's my thumb in the picture next to one of the newly hatched nymphs. As they get a little bigger, they have this bright red coloration. We usually see that around mid to late summer, but they still have those white spots on them and some black patches. Then as adults, the life stage most people are more familiar with. They have gray-brown spotted wings with some like lacy patterns at the very end of it. And you can see there those red spots that are the antennas that Astra was mentioning earlier. They just look like little red dots on their faces. Their bright colors are actually hidden when they're at rest, which is how you're going to typically see them. But If one is about to fly or is dead, or if someone, like I did in this photo, picked one up to get a picture of it and is holding on to it and it's desperately trying to fly away, you might get to see those bright colors. And those are a red-orange streak down their wings with black spots on it and a white swoosh, and then red at the tip. Now, the egg masses. This is the life stage, as we've mentioned in the last poll, that you're going to see at this time of year. They're each about 1.5 inches long and contain between 30 to 50 egg masses. They're covered in a grayish-brown putty-like coating. You saw some of that in the video. And then that coating is covering all these lines of eggs. So you may have seen some of that popping, the liquid inside the eggs that comes through when you actually crush them. That's what we're looking for there. Now, some of the egg masses are uncovered. It really varies, but most of them are going to be those covered egg masses that you see in the first two photos. Now, where can you find them? Well, pretty much everywhere. We can see them on trees quite a bit, particularly under sheltered bark. Sometimes if you have a dead tree and you peel off some of that bark, there'll be a whole bunch underneath, and you'll actually need to check both the trunk of the tree and then also that bark that you've peeled off on both sides, and you'll find egg masses on them. Also, firewood is a big place you can see them. Cars, other vehicles, furniture outside, camping equipment, rusty metal, cinder blocks, cushions, pretty much anything that's outside and is a relatively flat surface, there's potential for them to lay their eggs on them. So if you're moving anything from an area with spotted lanternfly to an area without spotted lanternfly, or really anywhere else, it's worthwhile to just give it a quick check over to make sure that there are no egg masses on them, or in the summer, no nymphs or adults. Now, there are some lookalikes. The first one is spongy moth, which is another invasive insect. The big difference is that spotted lanternfly egg masses sort of have a putty-like texture, or they look kind of like clay that's been just scraped onto something, whereas the spongy moth egg masses are kind of fuzzy and furry. Also, praying mantis egg masses can also be confused with spotted lanternfly, but here again, they sort of feel like little balls of styrofoam more than the spotted lanternfly egg masses, which are, once again, kind of like putty. Now, there are a whole bunch of different types of funguses and lichens and other things that can get mixed up with spotted lanternfly egg masses. Some big things to look for are the size. So if it's bigger than, about 1.5 inches up to 2 inches, probably not spotted lanternfly. If it's really thick, probably not spotted lanternfly, or if it's really, really thin and looks like it's just been spray painted on. If it has sort of like a leafy texture or it's curling up at the egg masses, may not be spotted lanternfly. But again, some of these funguses and lichen can look very, very similar to the spotted lanternfly egg masses. So if you're ever not sure, feel free to reach out to us. We're happy to take a look at it and give you some information about what it is. So how do you do that? Well, now we get into the reporting section. If you see spotted lanternfly or any other invasive insect that you're concerned about, snap a picture, note your location, and send in a report to us. That picture can be extremely useful for us. So what I always say is, particularly with the more mobile life stages, get that picture as soon as you see it. Don't try and creep closer or anything like that. Just get that photo immediately. And then if you want to get a little bit closer, you can do that afterwards. But if it moves away, at least you've got that initial photo. If you aren't able to get a photo, please do still send in a report. Those are useful to us even without a photo. It's just that photo is, it's an extra bit of evidence that helps us confirm the ID. So we'd prefer that, but it's not required. If you are not sure if it's an invasive species or something else, still send in a report. We are happy to get any good faith reports. If it turns out it's not an invasive species, all that's going to happen is we'll try to ID it for you and send you back a fun fact about it. We are bug people who are reviewing these reports, so we're perfectly happy to get those other photos and give you some good information about those insects. If you see something weird, so, you know, you're not sure it's an invasive species, but it's this insect you've never seen before, or a bunch of one type of tree are suddenly dying, still ask someone about it. That could be us. That could be your friend who's a naturalist. That could be an extension agent. That could be the local bird and nature club. Just check in with someone who might be able to help you figure out what it is, because that's how we often find new invasive species that we weren't even aware were in the country. A lot of times, you know, it's just going to be a native species that you've never seen before, but every now and then it is something of concern. So better to be safe than sorry with all of these. We also have resources. You can download those off of our website. We've got fact sheets and checklists, all sorts of things. If you want to make your own materials, we are happy to provide photos. All we ask is that you give us credit for the photos. And if you are either living or working in Massachusetts, we are also always perfectly happy to send you free materials. So that's our mini posters, flyers, stickers, ID cards. So if you want some of those scraper cards we showed earlier, we can send those to you. And you can get those either on that link that I've got above there or through the QR code in the corner. Finally, we have a newsletter. It comes out once a month. 5 bullet points or less, and it's timely forest pest information. We try to keep it short and sweet and then link out to further reading if you're interested. So again, you can sign up either with this link or the QR code, and we'll also put the link in the chat. And with that, we will now take any questions.
Thank you, Elizabeth. We have a lot of really great questions in here, and a lot of them are from Melody from her talk. So I think I'll try to address several of those first. So the first question that I wanted to address was one that came in when Melody was showing the Stockton slide. Somebody asked why the 4th instar population dropped.
So this work was done in a laboratory, and the largest cages that we were using were about 5 feet tall, 3 feet by 3 feet. And so that means that the largest living ailanthus and grapevines that we could put in there weren't huge. The larger the insect, including adults, the more food they require. So laboratory is not perfect, but in that particular graph, they didn't actually drop. They were just more spread out over time. They weren't as synchronous, so there wasn't 90% of them present all at once. The peak percentage at which they appeared was about 70%, whereas the first, second, and third instars were much more synchronous, and they were appearing at about 90% all at once. Same thing for the adults. They appeared over time, so there was no more than about 70% total present at one period in time, but the numbers were still there. So hopefully that helps.
Thank you. Somebody else had a question. Did I hear right that in Western Mass, for example, so a lot of us are here from Massachusetts, that the population of lanternflies would not sustain itself due to temperatures being like less than, well, it says less than 0.7 per adult, but maybe you could just speak to I saw a lot of blue out in Western Mass on that map you had up. Can you
Correct.
try to
Yes.
answer that?
So that map is an estimate that some of the modelers have put together at assuming all of the information that we know, how much the population would increase or stay the same or potentially decrease over time based on the effects of temperature, et cetera. And yes, in Massachusetts, given the right temperature conditions, particularly in the fall, winter, you could have adults dying early, you could have eggs dying and not hatching. And so the number of individuals present from year to year might stay about the same rather than build up larger populations. That's all that map was suggesting with the blue color.
Under ideal conditions, how long would you expect an adult lanternfly to survive?
Well, under the alternating regime for Stockton, California, the longest lived ones were like 100, 102 days. So if they're not being killed by fall temperatures, they can live a long time. I think the average was closer to 70 days. So not short.
Yeah, yeah, and I mean, Astra can speak to this, but I believe she was reporting that she was finding adults pretty far into the, like, close to winter, like really late fall into December last year. Is that accurate?
It all
Yeah, we
depends on,
definitely had adults here and there that were still alive after the like hard freeze that we had. Most of them died off, but you could still find a few.
after the 28 C or
Oh yeah.
28 Fahrenheit. Yeah.
Yep.
I'm not surprised there could be some adaptation going on.
Really interesting. Thank you. Someone was asking, and I guess this is for Melanie again. Like, is it possible that warming temperatures could kill adults before they have a chance to lay eggs?
It would have to be temperatures that we don't tolerate. And I saw there was some confusion between what kills adults without food and what kills them when they do have food. When they do have food, they can withstand up to 130 degrees as long as it's with cooler temperatures at some point in the day. But without food, they don't live more than a couple days at 86 degrees Fahrenheit.
Okay, thank you. Okay, somebody was asking about the blue on the map, and I did not make a note of which slide, so it might have been the model. And that might be a hard question to answer, but Melody, you tell me yes or no if you think you can take that one.
What was the question about the blue?
It just says, what was the blue? Sorry, I probably
Okay.
shouldn't have picked that one.
Well, the blue on the growth rate forecast map, that is where the population is not completely replacing itself from year to year. So it's not increasing in size and maybe not even maintaining the population size that it currently has.
Thank you. Some questions about egg masses that I'll probably throw to Astra, although other folks can feel free to weigh in. Is there like an average height where the egg masses get laid?
I mean, Melody might be able to speak better to an average height, but they will lay them from ground level to the tops of the trees. And I've heard from different research over the years that they like to go higher. But when we collect the data, they ask like how much, like how many do we find below 6 feet, so that they kind of know how many are low, like what percentages are low, and then what are high.
Thank you. Do you have anything to add, Melody?
Well, at the Fairfield site, it depended on the tree. Some trees had like 70% up higher, and 30% low, and vice versa. And I think it really depends on temperatures. Like I said, the adults will come down when they want to stay cool, and they'll come down if it's too cold. So they're going to lay wherever they're at up high feeding, if the temperatures are permissive.
Thank you. Someone asked about our treatment plans for this year. I mean, I don't know if they're finalized or how comfortable you are speaking to that, but anything you want to add, Astra?
We're hoping to do Dinoteph urine earlier in the season on Alanthus trees, and then we'll see how effective that is before we target sites again for bifenthrin in the early fall. Dinoteph urine is a systemic, and we're hoping that we can treat a bunch of trees kind of in the hot areas, and then when the adults kind of flock to those Alanthus to breed, we'll kill off a bunch of them that way. But we'll see what our adult populations look like after that, to do the bifenthrin treatments.
Thank you. This is a really good question. Why scrape the egg masses after popping? If you pop them, could you just leave them there, or is there like what's the reasoning there?
I think it's just to sort of be sure that everything's off the tree. There's some research that suggests like disturbing them like that will kill them. But I think Melody might be able to speak better to that.
Melody, do you have anything to add?
If you missed squishing some and you
Mhm.
just scrape them off, they could still hatch. So I think both is required, really.
Okay, so that kind of segues into another question about like, would you just scrape it off or do you have to collect scrape stuff? But that's why you guys are popping them, right? I mean, you did speak a little bit about the process of like scraping and into a container with like rubbing alcohol or hand sanitizer, but that I think that makes it a lot more laborious of a process than just popping the eggs. Is that accurate?
Yeah, I mean, that would just be more time consuming. And we're already popping the eggs. And when they're up high and we're scraping them with the poles, we're just trying to get them off the tree in hopes that, the mechanism of getting them off the tree will disturb them enough to kill them, or they'll get trapped under some snow. It'll be too cold for them, et cetera, or maybe some pests or some predators will get them. But it's more about just trying to disturb them as much as possible.
Thank you. Elizabeth, somebody asked about similar things to the SLF egg masses. Can you post a link to the lookalike sheet in the chat?
Yeah, absolutely.
you.
Yeah, because there are quite a few things, and like I said before, really, if you're ever not sure, we are more than happy to take a look and let you know if it's spotted lanternfly or something else.
Someone was asking why we don't just cut down and chip a tree that has a lot of egg masses. I'm gonna take that one. It's First of all, it's really, really expensive to do that. Second of all, the tree grows back, so it doesn't necessarily, it's a temporary resolution and it's tree of heaven, so it will sucker and produce a whole bunch more stems and just sometimes be even more of a nuisance and cause any egg masses you miss to then the nymphs hatch and just kind of disperse everywhere instead of just being on the one tree. Um, we did try that at first when we had very, very limited levels of infestation. And we had the resources and it wasn't effective. Somebody asked about, this is a tough question. I almost didn't put it out there, Erica, but let's do it. What are our long-term objectives with spotted lanternfly? I can say for Massachusetts that our goal is to slow the spread and potentially prevent new infestations. I think we found on a national level that this is not a pest that is able to effectively be eradicated at this point. Does does anyone else have anything to add? We could probably give a whole other webinar about this.
I think for Massachusetts, we're just trying to keep it out of agriculture, try to keep it from spreading as much as we can. It's kind of an uphill battle.
Folks that have specific questions about where something was found, like within a town in Massachusetts that has lanternfly, it's a little tricky. I can't tell, for folks that work for the town, please contact us. For folks that aren't part of municipal government, like sometimes we don't give out the exact location and you don't hear from us unless you're in the infestation. So you could try contacting the town, but a lot of the properties we find lanternfly on are private properties, so we don't give out the exact address. So that's an answer and sort of a non-answer. All right, somebody asked, are they strong flyers? And I think maybe I'll let Melody take that.
Okay. So all the published information about them suggests that they're not strong flyers, that they tend to glide more than fly. But I have seen them flying fairly well, from tree to tree and up higher around trees. So there's some question as to actually how strong a flight they are capable of.
Someone was asking about tree climbers. Does anybody want to talk about the issue about spotted lanternfly egg masses being laid too high up for the pole scrapers or anything, and what that means, and like why we aren't necessarily using tree climbers like we do for Asian longhorn beetle?
Sure. So we still are on the ground with binoculars recording all of the egg masses that we see on the trees, so that we know sort of what our populations are going to look like in the spring when hatch happens. And we target areas that have had like more egg masses that we couldn't reach, and stuff for trapping, so that hopefully when they hatch and they're coming down the trees, in some areas we'll put out bug barrier traps to catch those nymphs as they're coming down the trees and sort of mitigate the populations that way. But we do do a extensive survey of all areas, and we're looking at the trees and recording that data. So it's not like they're just going completely like unsurveyed or anything like that.
I have a comment that applies to that too. We did find some egg masses in our Fairfield site that were on branches that were less than a centimeter in diameter, so about 1/4 inch in diameter branches. And so be hard to even scrape those, and a tree climber couldn't even get close to something that small.
Yeah, we definitely see those as well. And like having tree climbers, I mean, I think that's just more staff. We need like funding for that as far as like ALB goes. You know, they have a federal quarantine and they have a lot of staff at the DCR that are already doing tree climbing for that. And it's also, I think, just easier in that respect to like go up and check, and like Melody said, like they can see where those exit holes and that and those you know, egg laying areas will be, because they're going to be on wider diameter branches.
And I'm going to take one more question before I close things out. And that is, somebody was asking about using a stiff brush to pop and scrape the eggs because it might be good to get into the grooves of the bark. So I figured I would just throw it out there for folks to answer.
You could. They do use like wire brushes, I think, in some states. I find that there probably is a lot of places where there's an egg mass where you wouldn't be able to really access the full egg mass with that brush, and the thin scraper card works the best. Or like in some cases, I just pick a sturdy stick up off the ground that will fit into whatever the notch is and kind of twist at it to pop the eggs that way. It just really depends on like where the egg is laid.
Thank you. And I think we should call it there. I want to again thank all of our speakers today, both in-house and especially Dr. Melody Kenna. Thank you so much for your presentation. And thank you, Astra and Elizabeth, for your updates as well. Thank you everyone for attending, and especially to those 100+ folks that stayed until the bitter end. We're going to post an archive of this up on our YouTube channel. You'll get an email with the link in about a week or so. And I hope you enjoy the rest of your Valentine's Day. Thank you, everybody.